Okay, sorry for inconvenience. Just I have to start.
No, no problem. Please continue.
Yeah. Uh, Good afternoon, sir, and my dear research scholars. My name is Santosh Rambhau Ippar, research scholar having a research topic or working on research topic, that is improvisation of phenotypic traits and disease resistance in chili varieties using double haploid technology under the guidance of Professor Dr. Nishikant Sadanand Lokhande, Department of Botany, Badrinarayan Barwale Mahavidyalay or Badrinarayan Barwale College, Jalna. My research center is also in Jalna, that is Jalna Education Societies, R.G. Bagdia Arts, ESB Lakhotia Commerce, and R. Besanji Science College, Jalna. Now, just as we discussed, chili is important crop because it is used in the spices, and chili belongs to the family Solanaceae. It is popularly known as hot pepper, red pepper, or chili pepper, and was originated in the tropical America. It is broadly cultivated in different countries, that is Britain, Australia, New Zealand, South Africa, India, USA, Central American, South American countries, and other Asian countries. Among all countries, India is the world's largest producer, consumer, and exporter of chili peppers. If you see its taxonomical data, the genus Capsicum is having more than 90 genera and 2,500 species of flowering plants, which includes different vegetable crops, that is tomato, potato, and eggplant. Now, this genus is native to tropical and subtropical America. If in case of history, we see, then the chili crop is introduced by Columbus in Europe in 15th century and has since spread to the rest of globe along the spice trading routes to Africa, India, China, and Japan. Specifically about our country, we can say, in India, it was introduced by Portuguese in 16th century. At the beginning, it was spread into the southern parts of India, and then overall or throughout India, it is spread at the end of 19th century. If here we can say India is the world's largest producer, consumer, and exporter of the chili. It is also produced, means chili crop is cultivated in the developed countries. For example, in USA, Canada, UK, Saudi Arabia, Singapore, Malaysia, Germany, and many other countries across the world. In India, major states which cultivate chili are Andhra Pradesh, Maharashtra, Karnataka, Uttar Pradesh, Punjab, Tamil Nadu, Rajasthan, Odisha, West Bengal, and Madhya Pradesh. In other states also chili is cultivated, but in less amount, we can say, means minor regions are under the cultivation of chili crops. Chili is having very good importance in case of diet, in case of economy. Here, this slide shows the fruit, means the rich sources of chili fruits. Means chili fruits are rich with vitamin A, C, and E, and also having minerals like manganese, potassium, thiamine, molybdenum, folate, and copper. Now, as we discuss the commercialization of chili fruits or chili seeds with 10 grams having cost about 500 rupees. In case of fruit cost, sometimes fruit cost increases up to 200 rupees per kg in India also. Means with commercial value, it is having very important value in the diets. Means chili fruits are having pungency due to capsaicinoids. And these characteristics are not present in all vegetable crops. In also capsicum means sweet pepper. Capsaicinoids are present in very less amount. So it is called as sweet pepper. And if pungency is more in chili, that is due to capsaicinoids, it will be called as hot pepper. Now, if we see the regions where chili is cultivated in India, the area under green chili cultivation in India is 0.309 million hectares with a production of 3.592 million tons, and under dried chili is 0.752 million hectares with a production of 2.149 million tons. Today, about 35 capsicum species are reported, 5 of which Capsicum annum, Capsicum baccatum, Capsicum chinense, Capsicum frutescens, and Capsicum pubescens have been domesticated and widespread with different terms depending on the region of cultivations. Means if Capsicum annum is cultivated in Maharashtra, then there is no need of cultivation of Capsicum annum in other states like Andhra Pradesh. In Andhra Pradesh, there might be cultivation of Capsicum frutescens or Capsicum pubescens depending upon the different environmental conditions. Okay. Then here you can see homozygous plants has a significant impact on agricultural system, which are nowadays have become integral part of the breeding program. Means in breeding program, the time is required very huge. Means time consuming is more during the breeding program. While in vitro, we can develop homozygous plants within a short period. Now, here is one research group, that is with Guha and Maheshwari researchers made the first report on pollen embryogenesis by anther culture of Datura inoxia. Means here we are discussing about the homozygous plants, means parental lines, because my research topic is development of some characteristics in chili plants by using double haploid technology. Double haploid technology means the development of homozygous plants. Means first part is development of haploid plants, and then we have to diploidize it from haploid to double haploid or homozygous plant. Here, the production of haploids and double haploids through gametic embryogenesis allows single-step development of complete homozygous lines from heterozygous parents, shortening the time required to produce homozygous plants in comparison with the conventional breeding methods, with carrying out several bracт crosses and many generations of selfing, which are time-consuming and labor-intensive procedures. Means traditionally, when plant breeders were doing their experiments on the field, these experiments will be needed labor and more time. But in vitro, no labors are required. Researchers also can perform their experiment with less time. The first studies of anther culture of Capsicum annum were reported in 1973 using semi-solid media. Then pepper is accepted to be the third solanous crop that could be defined as recalcitrant in regard to response to androgenesis induction. Now, why we need to develop such homozygous plants within a short period? There are some macro objectives, means major objectives of breeding also, or in vitro development of parental line also. So here first is the main agronomic traits such as yield, fruit features such as color, shape, plant habit, and fruit set. Second one is the resistances to abiotic stresses such as drought and salinity, which limit the cultivation in certain areas. Third one is the resistances to a plethora of bacterial, means huge amount of bacterial, fungal, and viral disease causing severe damage to cultivations and loss of quality of the production. Fourth, that is last one, is the quality, for which breeding objectives are mainly related to the improvement of various bioactive compounds such as capsaicinoids, isoprenoids, flavonoids, and vitamin C. Among these characteristics, other means other than these four objectives, another characteristics are also needed to improve in case of chili, that is fruit typology, fruit colors, pungency, qualitative traits. Then double haploid technology used to develop, means major outcome of the double haploid technology is less time required. Then second is the purity, that is the 1 to 2 years we required for the development of parental lines instead of 6 to 8 years required by the conventional breeding. In case of in vitro development of parental lines, we can develop parental lines with the 100% purity, which will be 99.2 when we develop parental lines with the conventional breeding. Now, this slide shows the literature review, means some researcher already worked such type of topics. That is Sanchez first literature, that is Sanchez, Conorado, means two different researchers work for obtaining the haploids and double haploids in cultivars of capsicum species. Then second review, that is Lakshmi, Sahitya, Sri Dipti, Kasim, Sunita, then Krishna. This second review, sir, am I audible?
Yeah, no problem.
Okay. Then second review.
Please tell the aim of that, whatever reviews you are taking, no?
Yeah.
Yeah.
In first review, these authors obtained haploid and double haploid cultivars, means how we can obtain haploid and double haploid plants or cultivars from the capsicum species. They described and impact on the genetic improvement of this crop due to this technology also described. Now, in second review, these authors focused the disease control strategies or plant pathogen interactions, then anthracnose disease and causative organisms which occurs on the chili crop, and also described
Yes.
its management. Then
Yeah.
in
Have you
third,
mentioned the results of that?
no sir, only what they focus that I have mentioned here.
Yes, and even the methodology, no?
Yeah, some methodology. Yeah.
Some methodology. Okay.
Oh,
Go ahead.
yeah. Then third review, these researchers, Shumilina and Shamkova, they shown here how the methods of isolation work out in the haploid and dihaploid paper plants in case of anther and microspore culture. Then they also focus which factors are having effects in case of embryogenesis, in case of paper microspore and anther regenerations. Then this is very important research article, which is of Supena, Soharsono, Jacobsen and clusters. These authors or these researchers explained one protocol, that is shade microspore culture protocol for the development of embryos, then plantlet from the microspores. Here is one method, that is they isolated anthers which are having 50% late unicellular microspores and inoculated in this shade microspore culture media, means double layer medium they used here. One is semi-solid medium and another one is the liquid medium to create some conditions required for the development of embryos in petri plates in aseptic conditions. So they isolated anthers, then inoculated after sterilization in the petri plates which containing double layer medium, incubated in different conditions, that is before inoculation 1 day at 4 degrees Celsius, and after inoculation, 1 week at 9 degrees Celsius, and remaining period of incubation used was 28 degrees Celsius in continuous dark. Here is the media composition they use, that is 2% maltose, 1% activated charcoal, then solid underlayer and 2.5 micromolar G18, 5 micromolar indole acetic acid in the liquid upper layer. Means both media used in the same petri plate. Below layer is semi-solid and upper layer is a liquid. Yeah. Then these again two research articles. First is the Wang Sun Chen. These explain first report, or they first time studied the anther culture of paper, were reported in 1973 using semi-solid media. Then in 1964, Guha Maheshwari, who did his experiments on the plant, that is Datura inoxia, for they used Datura inoxia for the embryogenesis from anther culture. Now, here is the scope of my research work. Means what type of scope is related to the chili double haploids. Here we can see there are different qualities of fruits and different size, shape, colors. So with the double haploid technology, we can develop different colored fruits, then different plants which are having disease resistant capacities. Now, this is the relevance of our research work. Means we are having relevancy towards our research topic because of need of more production, more production of chili according to the need of population. There has lately been a trend worldwide to emphasize over the production of non-staple crops, particularly horticultural and medicinal crops. And as these have a great potential for the economic importance. These are our significance of proposed problem, that is this research will demonstrate expression of recessive traits. We are well known from the Mendelian genetics, that is if two homologous chromosomes are there, if at one local means in one chromosome, if capital T is there and at another chromosome there is a small t, then dominant characteristics are expressed, that is with the capital T, not with the small t. But by using this technology, the recessive traits also can be expressed, that is small t, by after the diplodization of haploid plants. This research will help to develop homozygous lines in short period instead of long period, that is 6 to 8 years. Then our investigation will produce homozygous lines with 100% purity. This will help to produce plants with improved traits according to market value. This work will reduce the cost of pesticides for farmers because of the development of disease-resistant plants. Our work is significantly save environment from air and soil pollution, which will be caused due to pesticides in future. Now, these are our objectives: survey of various diseases and phenotypic traits of chili varieties, to collect chili disease resistant varieties from research institute like NBPGR, New Delhi and India, to develop 100% pure line for improved phenotypic trait and disease resistance using DH, means double haploid technology, in short period as compared to conventional plant breeding, to conduct field trial for checking improved phenotypic traits, and to conduct field trial for disease resistant in developed double haploids. Now, till date, we completed two objectives, that is survey and collection or procurement of chili disease resistant varieties from the research institute, that is NBPGR. And these
So,
three,
Santosh,
yes,
excuse
sir.
me.
Yeah.
Yeah, up till now, how many research centers you have visited for the collection?
One, sir, NBPGR, and I procured from two different, means from Hyderabad, Nainital.
Yes. So, are you in contact with them?
Yes, sir. Just before 4 to 5 days, I contacted with one lady that is in
Hmm.
Hyderabad. because I
Okay.
have faced some problems during the germination of seeds that was procured from them.
Okay, no problem. No problem. Proceed.
Okay. Now this is the hypothesis or assumptions regarding our research work. From the developed double haploid plants of Chile, uh, which will be the 100% homozygous. Developed double haploids will be with improved traits. Developed double haploids will be with disease resistance capacities. Developed double haploids will might be having improved phenotypic traits and disease resistance. Then in short period, that is in 1 to 2 years, we can develop double haploids. This is research methodology. Collection of chili disease resistant varieties from research institutes. Screening of chili disease resistant varieties for checking resistance level against virus on field. In vitro development of double haploids from chili disease resistant varieties. Ploidy analysis of regenerated plants by Ploidy analyzer. Ploidy analyzer is an instrument with which we can analyze the plant is having single set of chromosome or double set of chromosome. Then seed collection from developed DH0 generation means first generation, means the plants those developed in vitro will be DH0 generation, and from these plants we have to collect the seeds. Screening of DH1 progenies for homozygosity, chili disease resistance, and improved phenotypic traits on the field. So this will be the expected outcome of our research topic, that is new 100. Am I audible, sir? Yeah. Okay.
Yeah. Now you are
Uh,
audible. Yeah.
yeah.
It was paused.
This is our expected outcome of our research, that is new 100% homozygous pure lines of capsicum annum for improved phenotypic traits and disease resistance with short time period than the conventional breeding. Means we have to overcome some problems which are faced during the plant breeding. That is, purity will be more in vitro developed homozygous plants than the plants developed during conventional plant breeding. Then phenotypic traits can be easily we can get with the homozygous plants. Disease resistance developed by using double haploid technology within a short time period, that is 1 to 2 years instead of 6 to 8 years by conventional methods or conventional breeding. This is our presentation related to our topic. Now these are references and bibliography. Okay, thank you. My topic is over. So topic is open for discussion.
Okay. Here I want to ask one thing, that whatever means visits you have done, and the process of collection and separation, that screening is going on. So
Yeah.
why not you have put some photographs of that?
Actually, time period is only about 10 minutes, sir, to explain. I have
Okay.
also
So according to
alternatively, I have also that PPT, but it is having
Mhm.
72 slides, sir. This is only,
Okay.
yeah,
So
these are only
as
28.
per your time schedule, you have managed that.
Yes, sir.
So I was not knowing exactly what they ask to present. That's why I asked this question, that why you haven't put the slides over there of the photographs of all the material,
Yeah,
like.
yes, sir.
Okay. So some other participants, they can ask.
Yeah, Attar madam, are you having any problem or any doubt regarding my topic?
How many slides were there in all?
28, sir.
28. And the time taken?
Uh, I think much more time we spent for this seminar. Only 3 minutes remaining for stopping this.
Okay, as per your schedule, no? Okay,
Yeah,
time schedule.
yeah. According to Zoom app.
Okay then.
Yeah.
Okay then. Thank you so much. Wish you all the best for your
Okay,
future
okay.
work.
Okay, thank you, sir. Thank you for hearing with such a patience. Yeah. Okay,
Okay,
I'm
thanks.
going to start. Yeah, yeah.